Good evening, everyone. My name is Ulf Hannelius. I'm the CEO of Diamyd Medical. We are a Swedish diabetes company. We are developing drugs that focus on the underlying disease mechanism of diabetes. And today, I will focus on something very interesting. I will focus on something that we press released this morning. So we have clinical results from a phase 1, phase 2 clinical trial that look very positive, and I will show you those results because I think that's the most interesting thing here. And then I will present obviously the company and more what we are doing with both. We have two drug candidates. And before we go into the good stuff, this is something you need to understand. You have to do your homework. But we jump directly into the results that we press released. And I will explain what all this means. So this is the indication is newly diagnosed type 1 diabetes. And what we want to achieve with this therapeutic, which is a Diamyd diabetes vaccine, is to preserve the endogenous insulin production that the patients have when they are diagnosed. They still have about 10 to 20% left of their beta cells that produce insulin. If we can preserve this, it would have a huge benefit for the patients. And in blue here, these are our patients that have been followed for 15 months, and the dashed line here is what it untreated or a placebo-treated group of patients in the same age group usually looks like. So the natural disease progression here. On the x-axis, you have the remaining beta cells from baseline. So here you have 100%, here 0%, and this is time in months. So you can see over a 15-month period, our patients have on average gone down about 18% in insulin production, whereas the expected after 50 months is around 40 to 50% reduction in insulin production. If this is translated to a relative effect, it's around 50% relative effect compared to the untreated. And that's a pretty significant effect. If we compare to our previous studies, we have been all the way into phase 3 with this drug candidate with a different route of administration. Then it was administered subcutaneously. Now it's administered directly in the lymph node. Then the effect was around 16%, non-significant in phase 3. Now we're talking about something that looks like being around 50%. What's important here is also that this translates to something that is immediately clinically relevant. So this is a biological effect. We are measuring a marker that directly links to how much insulin the patient can produce. But it's important to also show that this has an effect on at least one of these two parameters. So HbA1c, that's the long-term blood sugar levels in patients. So it's a biomarker that measures how the blood sugars have been for the past 2 to 3 months. What you usually see is that from baseline, you usually see a gradual increase in HbA1c. The less insulin production the patient has, it becomes more and more difficult for the patient to manage their disease, to keep the blood sugars within a very small, narrow window. Our patients in the trial go down from baseline, and they actually are, at 15 months, they've gone down about around 16% in HbA1c. Comparing to it, usually have an increase around 50% or more. Sorry, it's not here, it's around 20%, a bit less than 20%. And then we look at how much insulin do the patients need to, external insulin. So all patients at diagnosis are immediately put on insulin injections for the rest of their life. And usually here, what it looks like is the longer the disease progresses, they need to increase their insulin doses in order to manage their blood sugars. Our patients go down at baseline, and after 15 months, they are about at the same place they were at baseline, compared to an expected increase around 50% or more in insulin doses. So we hit all the 3 major efficacy endpoints. And we also have immunology, not on all 12 patients so far, but the immunology we have, which is also published, where we compare to the subcutaneous route of administration to internal lymphatic. We can see when you give it into the lymph nodes, the immunological response is much stronger. It persists for a much longer time. And we can see that we create antigen-specific T cells that secrete anti-inflammatory cytokines when it when they come in contact with GAD, which is the active component in our drug, and one of the proteins that the immune system, for some reason, recognizes as foreign in the body. So this was press released this morning, and like I said, this looks very promising, and we can compare to all the data we've had before, and most importantly, there have been no serious adverse events in these trials or any other trials we've ever done with this drug. So we have a safety database with around 1,000 patients or more that have been in trials with our drug. So if you look at the problem, diabetes is obviously a huge problem. It's a global epidemic with 425 million people suffering from the disease today. About 10% of these are autoimmune diabetics, so type 1 and something called LADA. And that's our main focus. Diabetes is coupled to, on average, a 10-year reduction in life expectancy. For type 1 diabetics that get their diagnosis before the age of 10, this is actually a very recently published Swedish register study. If you get an early diagnosis, girls have a reduced life expectancy of around 18 years and boys around 14 years lower. So it's a massive problem, and the only drug therapy there is today is insulin, which was discovered 100 years ago. And this is obviously coupled to massive healthcare costs. About 12% of the global healthcare costs go to diabetes. Only 10% is drugs treating diabetes. The rest 90% are trying to manage with all the complications, transplantations, amputations, heart failures. So we have type 1 diabetes with the autoimmune disease where the immune system destroys the insulin-producing cells. This is more common in children, and it's an aggressive disease progression. So at diagnosis, you have a life-threatening disease and you're put on lifelong insulin treatments. Type 2 is about 90% of all diabetics. There you have both impaired insulin sensitivity. The body doesn't react to insulin the way it should. And the beta cells are gradually burnt out. They are overworked and they gradually also die. And we have two drug candidates, Diamyd, which I just showed results from, which is a biologic antigen-specific immunotherapeutic, and Remagen, which is a GABA-based oral therapeutic. They work on two different mechanisms in diabetes. So this is to preserve the endogenous insulin-producing cells by doing an antigen-specific immunomodulation, and this is to regenerate the insulin-producing cells. And together, they have synergistic effects based on preclinical studies as well. So on their own, they have very nice effects and have their own markets. But the nice thing is that if you combine these, you get synergistic effects. And together, they could really go towards something that is a really transformative treatment in diabetes. Our clinical pipeline looks like this. So our two products, Diamyd, going into newly diagnosed type 1 diabetes. We have two trials. The phase 1, phase 2 trial, which I just showed the 15-month results from, which is still ongoing. It will have a total follow-up of 30 months. And then we have Diamyd 2, a phase 2b trial, which has started recruiting since November last year in Sweden, Spain, and Czech Republic. And then for Remagen, we are starting up now in September a clinical trial in Uppsala University, looking at both safety, but also if we can regenerate existing the insulin production. And there is a investigator-initiated trial ongoing in University of Alabama, looking at both GABA and the combination of GABA and GAD, which is Diamyd, which was fully recruited this summer, and it will come with results next year. This is a completely new animation, and I heard there is no sound in this room, so I can run the animation. It's a short one. It's a premiere today, so it's texted, so you can read the text here. So I will be silent for 1 minute, and hopefully you can read the text, and then I will explain some. Or there's no text, actually. Well, there's an ongoing inflammation in pancreas, and what we are doing, we are focusing on the lymphatic system. So we are administering directly into a lymph node, which is the GAD base, so Diamyd, which has the active component GAD65, which is one of the proteins that the immune system recognizes as foreign in type 1 diabetes and goes to attack against. And we want to remodulate the immune system to understand that this is nothing strange. So it's taken up by dendritic cells, antigen-presenting cells, and it's presented to T cells that are then re-educated, and they become anti-inflammatory. They get an anti-inflammatory profile. They spread through the body, through the lymphatic system and blood system, and they also reach the pancreas and the draining lymph nodes from the pancreas, where they encounter GAD again, and there they start to secrete their anti-inflammatory cytokines, and this reduces the inflammation, the autoimmune attack on the beta cells, and thereby we can preserve the insulin production. The real voiceover is it's a nicer voice than mine, but this has to do. It was the premiere, so Diamyd again. We want to induce immunological tolerance against GAD65, and in this way preserve the endogenous insulin production. Our clinical proof of concept results show that we can do this. Operationally, we are focusing on the phase 2b trial, which is recruiting patients. It has recruited over 60 patients so far. It's a total of 108 patients. And we want to confirm the results that I just showed in the placebo-controlled double-blinded study. And business-wise, we are focusing on partnerships, so out-licensing. And to summarize, it gives a positive long-term biological, clinical, immunological effect. It's a simple treatment. It's virtually painless. It's less painful than a venous puncture. We have a big database with trials with more than 1,000 patients. The market for only newly diagnosed type 1 is around 1 billion or more. This could be then expanded to LADA, which is a bigger part of the autoimmune diabetes. And then obviously prevention, going into high-risk individuals, is another way to broaden the label. Especially with a good safety profile, you can go into healthy individuals as well. And we have a strong regulatory and IP protection. So we have orphan designation in the US. We have substance of matter in the US until 2032 and have several patents pending for both way of administration, biomarkers, different booster injections, etc. And quickly about our second drug candidate, it's called Remagen. It's a GABA-based formulation. So the point is to activate GABA receptors in the pancreas. We know that it's druggable. This has been shown that it can do it. Clinical effect is to regenerate the endogenous insulin production. Several groups in the world using different preclinical models have shown that GABA actually does this. We are focusing on the trial that starts this month in Sweden, and we want to confirm the preclinical results and translate this to clinic. And here as well, we are focusing on partnerships. And again here, a lot of research showing that GABA stimulates regeneration of insulin production. It's an endogenous substance, which gives it a very good clinical safety profile. The market, only if we look at non-insulin-based drugs, it's around 20 billion or more, and it's growing. Obviously, if you come in with regenerative therapeutics, this market would grow quite considerably. And this will follow something called the 505(b)(2) regulatory pathway, which means since GABA is a known substance, we also have licenses with combinations with GABA receptor modulators that are generics. It will follow this pathway, which means that we can use all the knowledge that is out there, all the data that is out there around GABA and these modulators, which would mean in practice that after our 2 clinical trials that are ongoing, we would probably only need 1 pivotal trial after that before we are on market, given that everything looks good in those trials. So in summary, 2 drug candidates in clinical development, quite fantastic results from this one now, and quite exciting to go into clinic with this one as well if you can show insulin that it stimulates insulin regeneration. And the vision of the company has always been to cure diabetes. This is a long-term vision, and hopefully we will achieve this one day. Thank you.